Dr. Hines is a pediatric critical care physician and a vascular biology researcher at Wayne State University. He earned his MD and PhD in pharmacology from UNC Chapel Hill, and he's really done some extremely novel research on the functional attributes of sickle cell anemia, and we're very excited to have him give this presentation today in the research that he's doing and application of the BioFlux system to that research. For those of you that are listening in to the presentation, thanks again for participating. You can feel free to put questions in via the chat message boxes. We'll also have a question and answer session at the end of the talk, so please stay online for that at the end. And with that, once again, it's my honor and pleasure to introduce Dr. Patrick Hines, who's going to give the talk today. Thank you, Patrick.
Okay. Well, Jeff, thank you very much for that introduction, and I'd like to say good morning. There's still morning to everyone on the East Coast as well. And I'm excited to talk to you today about the work we've been doing, both through my academic appointment at Wayne State University and at Children's Hospital of Michigan, and as it's evolved with functional fluidics, a startup that we developed that we're using as the platform to do a lot of our contract research testing using the platforms that we've developed in the lab. So the title of the talk is Can Microfluidic Flow-Based Diagnostics Personalize Therapy in Sickle Cell Disease, a Vision for the Future? And I put this together specifically for the for this webinar. I thought this was a good platform to really focus on the testing platform as it relates to sickle cell disease, because so much has changed over the years. And I want to give a little bit of context, because I know not everyone's a hematologist or a sickle cell expert. I want to give a little historical context, and I think that'll put in a better perspective where we are today and why I think the microfluidic based platforms have so much potential, specifically in sickle cell disease. So can everyone hear me okay? Okay, so I'm not hearing feedback, so I'm assuming everyone can.
Yeah, you're coming through loud and clear.
Perfect, okay. So sickle cell disease was first discovered in the United States in 1910. The first account was a gentleman who arrived in the U.S. in 1904, who had developed respiratory symptoms and had an ongoing, recurrent bouts of these respiratory episodes. And when he arrived in the U.S. in 1904, he was a dental student, and he initially was in Chicago. He, during one of these respiratory episodes, got so sick that he had to go into the hospital, and it was during that admission where the medical intern who was taking care of him actually discovered that on a blood smear. And for any of you who are clinicians out there, imagining a medical intern in 2016 actually doing their own blood smear, taking it to the lab, looking at it under the scope, and making and diagnosing a new disease, that's almost unheard of. But that's actually what happened with sickle cell disease back in 1910. James Herrick, pictured here, was credited with that discovery of the first report of sickle cell disease in the New England Journal of Medicine in 2010. In 1910, and I split the history of sickle cell disease research and development in the U.S. into 4 phases. So this first phase I sort of think of as the observation phase, where we first made the initial observation, described the characteristics, the morphological cellular characteristics, and described the clinical manifestations of the disease. In 1949, landmark discovery by Linus Pauling, who discovered the molecular basis for sickle cell disease, which was the single amino acid mutation that resulted in normal hemoglobin or hemoglobin A becoming hemoglobin S, which characterizes sickle cell disease. And during this molecular description phase, there was a wealth of research focused on understanding the dynamics and the kinetics of the sickle hemoglobin molecule, and understanding what regulated this sickle hemoglobin and allowed it to form these long chains, which cause the cell of the sickle, which I'll illustrate a little bit later. So a lot of this research was happening during this yellow phase here. The green phase represents the phase somewhere between the 1980s and the 2000s, where non-sickle cell based basic science really exploded during this time. And there was a lot of research looking at unique adhesive characteristics of sickle erythrocytes, which really brought in people from beyond just hematology who were interested in inflammatory disorders, oncology, cardiologists, people from a lot of different backgrounds. And I think this is where this was a real driver in research and discovery in sickle cell disease during this period. Sort of the multidisciplinary approach that was taken once the characteristics of sickle cell disease became, in terms of the fundamental cellular processes, were understood to be more than just a single gene mutation in hemoglobin. And beyond 2000, I tend to think of as the multi-target therapeutic stage, where there was many of these basic science studies began to culminate into actual therapies that began to be studied in the preclinical and clinical context. In 1998, we had the first FDA-approved drug for sickle cell disease, hydroxyurea, which is essentially a repurposed chemotherapeutic drug previously used for leukemia that was found to upregulate the production of fetal hemoglobin in erythrocytes, and this has been probably one of the greatest achievements in sickle cell disease to date. However, it's the only FDA-approved therapy that we have for the prevention of sickle cell disease complications. So clearly we have a lot of work to do, but this is a really exciting time right now because of all of the new discoveries that are available. So stepping back a little bit, thinking about Linus Pauling again. So he figured out this molecular basis, and the way that he visualized this, these white circles here represent individual hemoglobin monomers, and under 100% oxygen, you see that they're all separated. As you decrease the oxygen saturation from 70%, 40%, 0%, you see more of these monomers starting to interact and become polymers, and these polymers begin to strand and ultimately form the sickled shape. And as we see in this series of photomicrographs that Linus Pauling actually took, we see the same cell which under more deoxygenated conditions actually takes on that sickled form. And not only is this a morphologic feature, but it affects the rigidity of the cell also. And these rigid sickled cells can damage the vasculature and have a more difficult time passing through the small diameter of the microvasculature. And that's why the circulation of these patients in the microvessels is a primary point of the defect that causes the organ damage that we see in sickle cell disease. This photomicrograph was actually taken from a recent PNS paper where we see that upon deoxygenating the blood, we can actually see it's not exactly real time, this is sped up about 10 times, but we can actually see the change in morphology of these cells just with deoxygenation. And so we began to do a lot of work understanding these hemoglobin properties, but as that was happening in the 1980s, we began to appreciate other properties as well. This is a seminal paper by Robert Hebel at the University of Minnesota, where he first described the adhesive characteristics of sickle erythrocytes. And he looked in a static assay where in a multi-well plate, there were cultured endothelial cells. and he plated sickle erythrocytes on these endothelial cells, washed them away, and looked at the ones that were still adhering. As you can see in panel A, the majority of these cells are actually normal in morphology. So morphologically normal cells, although they're from patients with sickle cell disease, clearly demonstrating a much higher level of adhesion than cells from an unaffected patient adhering to culture endothelial cells in this static context. So this really exploded an area of research where people began to try to better understand these adhesive interactions. This is the cover of a 2002 edition of Blood, where they illustrated what was known up to that point about the specific adhesive interactions that mediated the sickle erythrocyte vascular endothelial cell and subendothelial interaction between these sickle erythrocytes. And as you can see, there's a lot of different interactions. And fast forwarding to today, many of these interactions have now been exploited as therapeutic targets that are currently in clinical trials. So when I began studying sickle cell erythrocyte blood function back in the late 90s, one of the questions that I had was, given the episodic nature of sickle cell disease, the cells, if adhesion is a fundamental part of the disease, then the body must have some way of regulating the adhesive state of erythrocytes. And this was a new way of thinking about erythrocyte function at the time, because platelets were the primary anucleated cell that was appreciated to have a very robust response to the environment, so that they could increase adhesivity at specific times when it needed to form a clot, and be relatively quiescent during times when it needed to maintain normal rheology and circulation. So what we discovered was that epinephrine and other beta-adrenergic agonists were actually able to increase in a very rapid way the adhesive properties of sickle erythrocytes, and we found that this happened through a cyclic AMP mediated mechanism. More specifically, when we looked at the baseline adhesion of about 50 patients, first of all, you see that there's a range of adhesion levels in these patients, and we believe that these different baseline adhesive properties really inform some of the baseline pathology that we see in these patients. And being able to understand this from a more clinical context may give us clues into which patients are at more risk. But when it came to the stimulation of these cells, when we treated the cells with epinephrine and we normalized that stimulated adhesion to 100%, we also saw that we were able to inhibit these stimulated cells with pre-treatment with common beta blockers such as propranolol. We also showed that if we inhibited protein kinase A in these cells, that we were able to inhibit this stimulated adhesion as well. And we were also able to inhibit this adhesion with a soluble inhibitor of bcam-lutherin, which was the molecule that was shown to be the supporter of this epinephrine stimulated adhesion to the extracellular matrix. So these studies were continued, and we were able to validate this in animal models. And Marilyn Tellen and colleagues at Duke University did the first clinical trials looking at propranolol as a potential anti-adhesive therapy in sickle cell disease, where they looked at a cohort of 5 patients in one cohort with a 10 milligram single dose, a cohort 2 with a medium dose, and a larger dose in a third cohort. And I thought it was really interesting the way they did this study, because they looked at patients who had a stimulated response to epinephrine. So that's another important feature of the disease, is that not all patients respond the same, both in terms of baseline adhesion and in the context of stimulated adhesion. And so her group specifically looked at patients that did have this stimulated response. And in our hands, this was about just under 50% of patients which respond to epinephrine in this way. And looking at patients that had this response, they went on to treat patients with these three concentrations of, with these three doses of propranolol, took blood from these patients, stimulated with epinephrine, and then looked to see how much inhibition they would get when patients were actually treated with the beta blocker. And they saw a nice dose-dependent decrease in adhesion in patients who took a one-time dose of this beta blocker, which is a really exciting example of basic science observations being taken from literally from the bench to the bedside in the context of sickle cell disease. So this sort of changed our way of thinking about the disease, where before people thought of patients being in a non-adhesive state when they weren't in crises, and being in a persistently adhesive state when they were having chronic pain or acute pain, acute chest, or any of the other manifestations of sickle cell disease. What we argued was a model where the patient exists in a quiescent state. They're exposed to some stimulus, and there's lots of studies now showing that there are many inflammatory mediators beyond just epinephrine that can put these sickle erythrocytes in a pro-adhesive state. So there's an exposure to a stimulus that increases the adhesive properties of these cells. And now this patient is in a crisis state where these cells are much more likely to participate in adhesive interactions with the microvasculature, and much more likely to actually occlude the microvasculature, causing downstream problems with circulation. And the patient cycles from this stimulated state into a more quiescent state, going on to get subsequent exposures to a stimulus, which causes the cycle to take place again. So this then causes us to think about a window of opportunity. If patients cycle between this activated state and this quiescent state, then it would be really helpful if we could identify which state the patient is actually in before they actually show up in crises. Because the way sickle cell disease management, or any disease which is characterized by acute vascular obstruction, such as myocardial infarction, stroke, we tend to intervene at this phase. And as a pediatric ICU physician, I can really appreciate the fact that this is where our real clinical expertise lies. How do we manage patients once they come to us with an acute obstruction? But there's a real opportunity here if we can figure out what are the characteristics of these cells before this acute obstruction takes place. Are there changes that might take place that we can identify and that will give us a window of opportunity to use other treatment options that might prevent the patient from cycling to this acute crisis state altogether? And now that we're in this period where there's lots of therapies being studied for sickle cell disease in a clinical context, such as hydroxyurea, which is already approved, and now we're looking at much more broad applications of it, inhibitors of selected molecules, which have been shown to play a role in these pathologic interactions of sickle erythrocytes. Beta blockers, VLA4 inhibitors have been studied. Inhibitors of hemoglobin polymerization have been studied as well, as in terms of their ability to improve rheology. There may be a window where we can deploy these therapies before the patient presents with any clinical symptoms, if we can define this period on a biologic level. And there may also be an opportunity to deploy some of these inhibitors in the acute context as well. But we're particularly interested in ways that we can prevent this from happening altogether. So when we think about ways that we do this, we have to step back a little bit and think about, so what are we trying to to, to, to mimic? And this video represents the type of blood flow that we see in a post-capillary venule, where we still see the pulsatile nature of this blood flow, and we see how this blood is flowing through these small vessels. We're really trying to predict what this blood is likely to do. And the way that we've done it up to this point in sickle cell disease is using ultrasound type technology, such as transcranial Doppler. And without going to exactly what these images represent, the bottom line is that when you look at ultrasound data from the cerebral vessels in a patient with sickle cell disease, we've shown that if there is normal transcranial Doppler flow, and here we see a velocity of 123 in a normal situation versus a patient who has high velocity, in this patient 271, there's been lots of studies to demonstrate that these patients tend to be at higher risk when we identify that they have high flow, which has been a tremendous achievement in sickle cell disease, where we can identify patients at risk of stroke and providing a window of opportunity where we can intervene in those patients with things like hydroxyurea and chronic transfusion therapy. The problem is, by the time that we see an increase in flow on something like Doppler, there's a lot of damage that's already taken place. And to illustrate that, this photomicrograph, which was not photomicrograph, but this illustration, which was taken from the Lancet Neurology back in 2006, shows some of the many processes that are involved in sickle cell vasculopathy. This is specifically looking at vasculopathy in the context of stroke and sickle cell disease. So we see red blood cells, white blood cells, we see free hemoglobin, we see platelets, much more than just an erythrocyte mediated problem. And here, this left part of the picture represents the early phase of the disease, where the blood vessel is relatively normal, the endothelium is relatively normal, but within the blood, there's a lot of changes that are taking place. We have cells which are becoming more adhesive and are interacting more with the blood vessel wall. The membranes are becoming more fragile, and they're releasing free hemoglobin. Free hemoglobin is sequestering nitric oxide and putting the endothelium in a more pro-adhesive, inflamed state, where you get propagation of adhesion, you get which causes smooth muscle proliferation. And this smooth muscle proliferation, the intraluminal inflammation, all acts to create a blood vessel that is going to show higher flow velocities on something like transcranial Doppler. So by the time we recognize on something like transcranial Doppler that there's a problem, you can see that there's a lot of there's a lot of pathology that's already taken place, some of which is relatively irreversible. So right now, the clinical standard is that we intervene at this point. It would be much more advantageous to figure out a way to identify these risk factors in terms of blood function, where we can intervene back here, where we can see these problems that can occur within hours to days, as opposed to dealing with problems that have developed over weeks to months to years. So there lies the argument for a blood function diagnostic tool specifically for sickle cell disease. And the characteristics that I would say would make a diagnostic tool useful is that it could be used to predict the likelihood of microvascular circulation problems in these patients. It could be used to select which therapy is more likely to benefit blood function in an individual patient. And then once you've used this platform to select a therapy that the patient's blood will respond to, then you can use that same platform to monitor this response over time, which is going to require something with small blood volume requirements so that you can do serial monitoring in a population of patients that are anemic to begin with. So we sort of take a step back and look at the tools that we've used for research in sickle cell disease. And back in the days when Hebel was initially doing the first adhesion studies to the culture endothelium, folks were using static adhesion assays where you would immobilize the substrate of interest to a multi-channel plate, and then you would add the cells, wash the cells away, and you would manually count the cells that were still interacting. The pros of the situation are that it's simple, it's low cost. You can do this at relatively high throughput, although the quantifying the cells that are interacting can take some time if it isn't an automated process. The cons are that it's non-physiologic. We've learned that the adhesive interactions are affected in a very real way by shear flow, and that the application of shear really selects for interactions which are much more likely to be clinically relevant. And the other con is that it requires I won't say a large, but a moderate blood volume to be able to occupy the surface of these wells and be washed away. So the lack of physiologic relevance is a real issue in that platform. We look at other research tools such as the parallel plate flow chamber assay, which is an assay I became very familiar with during my graduate school days, and it was one of the workhorses of the discoveries in sickle cell adhesion during a certain period of time. The way that we did these assays, we took a silicone gasket and we placed it on a surface of either glass or a small tissue culture plate, and we used vacuum grease to actually create an airtight seal between the gasket and the surface. The cutout in that gasket was where we immobilized a adhesive substrate of interest, such as laminin, thrombospondin, VCAM, or where we cultured endothelial cells. We put a chamber on top of that where we had an input port, an output port, a vacuum that would create a seal around this chamber, and we would have flow across this this cutout area, and we would measure the adhesion from there. The pros was that it was relatively low cost, and it allowed us to look at adhesive properties under physiologic flow conditions. The cons was that it was very cumbersome. It probably added about 1.5 years to my PhD training, but very cumbersome. Lots of problems with air bubbles when you're trying to create this tight seal. Very low throughput, so it would take a long time to do a lot of different samples and conditions, and because of that, it was more difficult because you couldn't do as many studies in parallel. The analysis of the data was confounded by time. And then, it was a higher volume requirement because of the size of the area where the flow was actually happening. So other tools that were used, intravital microscopy, which there's very fascinating data that has validated many of the drug therapies that are currently under study now using these intravascular microscopy mouse models. And here we see a dorsal flat window that's placed on this mouse, and you can use a microscope to visualize in situ the flow of blood within a specific vessel. And you can actually look at the response to drug in terms of these adhesive interactions, and you can actually measure flow. The pros of that: this is an in vivo model, very high physiologic significance in terms of the effects that you see. Clearly, the cons are that it's a very complex system. A lot of training is needed to do it. Of course, it's low throughput. You really need an in vitro assay to do, screening and patient blood testing, and it's very costly. But when we combine all of those needs that we had when we thought about a diagnostic tool for sickle cell disease, and we looked at the options that were available, we really liked the BioFlux system because it met the pros that we thought were most meaningful in in the type of test that we wanted to create. It was relatively simple. The blood volumes were small. We could do this at pretty high throughput, and we could create physiologic conditions within the microfluidic flow chamber that allowed us to look at these interactions under physiologic flow. So just as an example, and for those of you who are users, this will all be familiar stuff to you, but this is sort of how we specifically adapted it for the study of sickle cell blood function. We would load the sickle cell blood sample in one of the inlet chambers with a wash buffer in another, and we would flow our cells through the viewing window. Once we flowed for a fixed amount of time, we would switch to the wash buffer and wash away the non-adherent or non-specifically adherent cells. We could use a number of different adhesive substrates and do all of this under physiologic temperature. Because we can also control the oxygen tension here, that's another benefit in the system as well. And so these were the properties of this system, which made it easy to adapt for sickle cell blood function testing for us. So we used the BioFlux platform to then build a specific whole blood assay that would create standardized conditions to measure sickle adhesion. And what we've done is to apply this platform for clinical research. We found a significant need for something like this in the context of clinical research for sickle cell disease because the way it stands now, if a particular drug is being tested in a population of patients, the selection criteria for these studies generally tends to be that the patient has the documented hemoglobin SS mutation, that the patient's within a certain age range, that the patient hasn't been transfused, very general, non-specific selection criteria characteristics. But these drugs tend to be very specific in their action. The drugs that affect erythrocyte adhesion and target specific receptors, or even target the surface of the erythrocyte more non-specifically. And with many of these studies, what we see is a broad range of responses where we see patients who really, on a clinical level, didn't respond at all, and there are patients who had a very significant response. But generally, none of these trials are able to demonstrate a significant efficacy beyond 50% of the patients that were used. We believe that one of the reasons for this was that we were selecting patients that had very little pre-test likelihood of responding to the drug because of intrinsic characteristics of that patient's cells. And because we didn't have the tools, we weren't able to define this population more efficiently. So what we've done with our platform is to preclinically, and this top part represents preclinical studies, the bottom part represents clinical testing. So preclinically identify a population of patients with the disease of interest. We would then take blood from each one of these patients. We would place it in the in our blood function assay. treat it with various concentrations of the drug of interest, and stratify patients into non-responders and responders based on their response. And we've done 3 of these preclinical trials. There are 5 of them that we're hoping to do in 2016. But what we've seen from the experience of having done these assays is that there is a range of response to the specific adhesion inhibitors that are being developed. And at least preclinically, we could identify patients that whose samples showed a significant inhibitory response to adhesion under flow. And what we vision for the clinical research applications for this is using something like this to help us select the population that we're going to take on to clinical testing. Once we identify this population, we would look at blood function in the context of adhesion under flow under these standardized conditions in each patient prior to treatment with the drug of interest in the clinical trial. So we have baseline adhesion data. The patient's treated. Because this uses such small blood volumes, it's not difficult to do serial blood monitoring post-treatment. And because the study is also collecting a wealth of clinical data, we can correlate this clinical data to the adhesion data and better understand what the relationship is between the actual inhibition of the adhesive properties of these cells and resolution of clinical symptoms. And so this is the clinical application in clinical trials will start in 2016, and hopefully we'll have data from those trials by the end of the year. So this really takes the one-size-fits-all model that currently exists for sickle cell disease, where when we think about the acute management, for example, the patients present in acute vaso-occlusive crises and pretty much get the same supportive management that they got 50 years ago: hydration, oxygen, analgesia for pain, to get them over the acute crises. When it comes to more chronic management, because we now have a FDA-approved therapy that prevents a lot of the complications of sickle cell disease, we're able to identify populations of patients that are defined by the severity of their disease. And hydroxyurea now is becoming much more of a standard of therapy, where even kids are being started on hydroxyurea pretty early. But it's likely going to be used pretty much across the board and in all comers. And we really aren't actively using a personalized medicine approach to identify patients likely to have a more or less beneficial response. However, in as little as probably 5 years, we're going to have lots of other options that providers are going to have to decide between, both in chronic management and acute management. And it's going to be very difficult for providers to figure out which therapy they should deploy for what individual patient. And that's where I think a platform like ours has a lot of potential value, is in helping providers at least understand preclinically what drug is likely to have a beneficial effect on adhesion in an individual patient. And we're in the process of collecting data to determine whether this type of data actually reflects or predicts the clinical response to a particular drug. And basically using this to, again, stratify patients and identify patients that are going to go on to be treated with a specific treatment approach. So just to get into some examples of preclinical studies that we've done with some of our pharmaceutical partners. Natalizumab is a anti-VLA4 monoclonal antibody that's currently approved for multiple sclerosis and Crohn's disease. Because it specifically targets the VLA4 integrin on the surface of leukocytes, the mechanism of action in the context of multiple sclerosis and Crohn's is that it prevents the stratification of leukocytes into sites of inflammation. In the context of multiple sclerosis, that would be a neuron, and prevent some of the ongoing damage and demyelination to neurons on a chronic basis. However, in sickle cell disease, we know that VLA4 also mediates the interaction of both leukocytes and reticulocytes to the vascular endothelium. So we asked the question of natalizumab binding to sickle reticulocytes and leukocytes would potentially block adhesion under flow. And so we did one of these preclinical studies where we, here I'm showing the data for the culture endothelium. And this data, I forgot to mention, was actually presented at the American Society of Hematology in 2014. So we cultured endothelial cells within the microchannels of the BioFlux plate, and we used flow cytometry to confirm that under stimulated conditions that we were able to upregulate adhesion receptor expression on the surface of the cell. We were specifically interested in VCAM upregulation, and here we see that we significantly increased the VCAM expression under these TNF-alpha stimulation conditions on our cultured endothelial cells. And when we did a dose response where we treated the patient blood samples, we see that we were able to inhibit adhesive interactions to the culture endothelium under physiologic flow conditions. So as we do with many of our targeted adhesion inhibitors, we validate the inhibition to the culture endothelium before going to a more isolated substrate of interest. And we're able to show this same pattern as well in the context of adhesion to isolated VCAM. We did a similar study in partnership with Mass Therapeutics where we looked at Veploxmer, and we just recently presented this data at the American Society of Hematology in Orlando this past December. Just for a little background for those who aren't familiar with this particular compound, Veploxmer is an agent that non-specifically engages the surface of these cells, and it reduces high surface tension adhesive interactions that can lead to more non-specific adhesive interactions between circulating cells and the endothelium, the vascular endothelium. So Veploxmer can reduce this surface tension and prevent some of these non-specific hydrophobic adhesive interactions from occurring. And this drug is actually in phase 3 trials in sickle cell patients right now. And what we did was identify 10 patients that, and we looked at blood samples from those patients, treated it with increasing concentrations of Veploxmer to look at the effect of this inhibition over time. And as we've seen in most of these situations, we have a range of baseline adhesion levels to begin with, and also a range in response to drug, as we see with this dose response. Although, despite that variation, we still saw statistically significant inhibition in adhesion even at our lowest concentration of Veploxmer used. And when we do a scatter plot so we can actually better visualize each individual patient, what we were particularly interested in is being able to identify this group of patients that has has less response to Veploxmer in the context of the standardized adhesion assay. And we think this is going to be very valuable in targeting therapy to patients that are most likely to have a clinical benefit. and the clinical validation of this is where we're putting a lot of our research efforts right now. So in closing, we're very interested in, although our focus is on sickle cell disease right now, and both developing a standardized adhesion assay that we can use to both define the, understand the baseline adhesion characteristics of erythrocytes for these patients, understand how these baseline adhesion characteristics correlate with their clinical risk, similar to the way that we're using transcranial Doppler now. We also want to see how this can help us better personalize medical therapy in sickle cell disease, particularly as it relates to the anti-adhesive agents and agents that are meant to improve rheology in these patients. So that's really our approach to thrombosis conditions and platelet function defects, bleeding disorders such as hemophilia, biofilms, various malignancies, and better understanding the adhesive characteristics of circulating tumor cells. Am I still on?
Yes, you're still on.
Okay, okay. And so there are lots of other potential applications that we'd be excited to partner with other collaborators to better understand how we can apply this microfluidic based platform to other clinical applications. So with that, I'll stop and take any questions from the audience at this time.
Patrick, this is Brian Haynes. I've got one question to start you out with. How complex do you think these adhesion assays can demonstrate in terms of different patient responses when you start getting into combinations of therapy instead of single therapy treatments, things that may take 4, 5, 6 treatments? Do you think you're going to be able to correlate at that point?
That's a really good question, Brian, and I think that it's going to be very important that we investigate whether we can do that, because combination therapy is on the horizon. Right now, these a lot of the companies that are developing these approaches are obviously validating them as single therapies, but no one target is going to be a silver bullet. Kind of how I illustrated in that earlier photo, Mike, that earlier cartoon. These adhesive processes are multifactorial. They're not only are you looking at adhesion, you're looking at stimulation and activation of the vascular endothelium. You're looking at activation of prothrombotic pathways. No one target is going to be the silver bullet to improve rheology in all patients. So just like with cancer therapy, the next 3 to 5 years, I mean, we're going to be shifting from single therapy to talking about how these therapies interact with each other, and we hope to have a database of response data that we can help to do some predictive modeling about this multi-drug response. But we think that it's very realistic to think that we can combine drugs and model that combination therapy in vitro and look at responses in that way as well. How predictive that's going to be in terms of the patient response is still left yet to be seen. But based on the single drug data, I would hypothesize that this would be a very valuable approach to better understanding and defining combination therapy as well.
We had a question that came in that and I know some of this is already in the publications that you have out, about what shear rate do you use and what volumes are required